Afternoon. My name is Parker Hoda, and this is my genetics extra credit video on replication, transcription, and translation. First of all, a little background information on DNA. DNA is the genetic material of all living organisms, and it's made of three primary components. It's made of a sugar, a phosphate, and a nitrogen-containing base. To better explain this, I'll draw it out so you can see what it looks like. So first, you have a 5' end and a 3' end. And there is another complementary strand of DNA that goes anti-parallel. It starts at the 3' end and runs to the 5' end. Alright, so your complementary bases are A and T and G and C. And they would be on here in any order. We'll go with A, G, T, A. The complementary strand would have the bases T, C, A. Now that we know what DNA looks like, we can look at the next step in the process, which is DNA replication. Alright, so DNA replication. First, in DNA replication, you will have something called an initiator protein come in right here, and it will open up the DNA so that it can be unwound by DNA helicase. So as you can see here, DNA helicase has already unwound right here, and it has DNA gyrase here in front of it that reduces the torque that happens so it doesn't get burnt. So you then have DNA primase, which lays down a nucleotide primer on a 3' OH group so that the DNA polymerase 3 can synthesize more nucleotides and make new DNA on the complementary strand. Alright, so as you can see here, there's a leading strand and a lagging strands. The lagging strands come afterwards, and they have to be bound by DNA ligase just in order for there not to be gaps. Now you have replication 4 strands of DNA. Alright, there's differences between prokaryotic and eukaryotic DNA. As you can see here, this is eukaryotic DNA, which is in the double helix form. Prokaryotic DNA is in a circular form, and so there are some differences, but for simplicity's sake, I'm just going to tell you all about eukaryotic. Some other differences are that eukaryotic DNA is much more abundant than prokaryotic DNA, and it also replicates more slowly. So there are more points of initiation in eukaryotic replication, whereas in prokaryotic there's only one replication origin. So as the story goes, DNA undergoes transcription and becomes RNA, and RNA undergoes translation and becomes a protein, and proteins are how everything works in the body. So let's go over how DNA turns into RNA through the process of transcription. Over here you've got your DNA, and this is a transcription unit, so this is everything you need for the process of transcription. So here you have your original DNA, and this is your RNA that's going to be formed. Alright, so within your transcription unit you have several regions. You have your promoter region, which is this first region here. You have the encoding region, and you have your termination sequence. Right here is your start site, and right here is your termination site. So what's going to happen are 3 different steps. You have the step of initiation, which is where your RNA polymerase and a sigma factor come, and they bind to the promoter region. It has to be recognized in order for the process to start. Then you have elongation, which takes place here at your encoding region, where these are the nucleotides that are actually transcribed into the new RNA. Then, once it gets to the termination sequence, begins the termination part of the process. It'll be a sequence of nucleotides that'll be recognized as saying this process needs to stop. And then it will be terminated at this spot, and you'll be left with a strand of RNA. And I forgot to mention that you have 2 strands here. One is going to be your template strand, and one is going to be your non-template strand. Your template strand is going to be the strand that is actually encoded into the RNA. So now that we have transcribed our DNA into RNA, we will translate our RNA into proteins. And the way that works is through the process of translation. So translation, like transcription, has 3 stages: initiation, elongation, and termination. Initiation in this process happens with this guy, Mr. Ribosome. Ribosome has a small subunit and a large subunit, and they bind to the mRNA that we got from transcription. So how this works is the mRNA flows through the ribosome, and the ribosome reads the mRNA in codons. Codons are 3 nucleotide sequences, and the ribosome finds the appropriate matching tRNA located here and conveniently shaped like a T. The tRNA has an amino acid hooked to it, and it also has an anticodon, which would be what matches. Okay, like I said, each tRNA is attached to an amino acid, and there are 20 different amino acids in the body. So the tRNA comes in and it binds to whichever codon fits its anticodon. When it does that, it brings its amino acid, and then the next codon gets its tRNA paired. And when this happens, the amino acids are eventually hooked together, and you get something that looks like this, a long chain of amino acids, which makes a polypeptide chain. The polypeptide chain then makes a protein. That's it.